Welcome to the second episode of the Nervous Breakdown podcast, where we post content.
Today, we're going to be looking at the innate immune system. But what is the immune system as a whole?
Well, the human immune system is the most complex system after the human brain, and it's a network of cells, tissues, and organs that fight against pathogens, and it is comprised of adaptive and innate immunity. Now, what is the difference?
Difference comes in the specificity of the responses. The innate response is non-specific for a wide variety of pathogens, whereas the adaptive immune system is specific for a certain type of pathogen, really focusing on the specific antigen on the pathogenic surface.
Okay, so let's dive straight into innate immunity. What are some of the physical or chemical barriers that protect us against pathogens?
So the innate immune system has lines of defense. The first line of defense is looking at these physical barriers. Now, these barriers can either be the physical ones of skin, um, epithelia, with their different characteristic properties to really stop pathogens getting into the tissues of the body. So things like tight junctions to really hold these cells together and stop pathogens from entering through is a key one. Then we have your um, secretion type defenses, such as mucus to trap pathogens to be moved out by the cilia, and also things like tears, which have lysozymes to break down different bacteria, the extracellular walls, things like that.
Hmm. So lysozymes are enzymes, right?
Yes, yeah.
And in fact, they come from the Greek word lyso, which means to break. And this is actually going to be important in many other processes we'll talk about, where we're trying to lyse, for example, the membrane, break it apart. So, now, what are the two, can we go through the two main lineages of cell
Yeah.
types in the entire?
So your innate immune system all is formed from white blood cells. These are the resident cells that will characteristically take up the effector functions and really do they will mediate the effects that the immune system functions for. So you have lymphoid and a myeloid lineage, which will form two different types of cell. Your lymphoid lineage will form your B cells, your T cells, and your natural killer cells. These all come from a common lymphoid progenitor. Now, it's important to note that although most of the lymphoid cells, your B and T cells, are in the adaptive response, the natural killer cells are part of the innate immune response, even though they are from the lymphoid lineage. Also, it's worth it to mention innate lymphoid cells, which again are from this lymphoid lineage, but they're in the innate immune response. From the myeloid lineage, you have your neutrophils, your macrophages, your dendritic cells, eosinophils, basophils. These are your more phagocytic type of cells, and is what their main role is within the innate immune response. They have other different effector functions, but they all resolve to undertake effects for a specific type of pathogen or a certain type of infected cell.
So, should we look into some of these different cells?
I think you should.
Okay, let's start off with neutrophils. So, evidently, as you can see, we're missing the sun.
And we're missing the cells.
So what are some of these different types of cells? Let's start with neutrophils. What lineage do they come from, and how are they formed as a process?
Neutrophils come from the myeloid lineage. Their common precursor is similar to your basophils and eosinophils as a myeloblast. These are one of the most abundant white blood cells, with over 70% of the white blood cells in your blood being neutrophils. They act as phagocytic cells, and they are present in your extracellular tissues. They are they receive signaling from different chemokines for where they should migrate to, and yeah, they are involved in your phagocytosis. They granulate and can also secrete their nuclear material to really trap pathogens and all what form your pus. So these are called your nuclear extracellular traps, and they are also involved with this degranulation process of free radicals, different enzymes, granzymes sometimes, to really break down your pathogens as well. And it's important to know that they die once they have done this.
Okay. Briefly, let's go through the three, I would say, least common or lowest in number. Is that true? Basophils, eosinophils, and mast cells. Are they low in number? How do they vary?
It's relative, but compared to the neutrophils, it's really very small proportion, maybe 5-10% each.
Okay. And what do each of these do, and how do their roles as well?
Okay. So these are your granulocytes. They are known for secreting granules. These granules will contain things like granzymes, which are sort of they have this catalytic ability, uh, reactive oxygen species, and all of these will actively break down cells. They can induce different apoptotic pathways within cells as well, but they really function to kind of break apart cell membranes, disrupt mitochondrial function of bacteria, things like that. The difference comes in the different type of responses they're used in. Now, these things, mast cells, are part of your anaphylactic response, so your allergic responses. They respond very well to IgE and they secrete histamine, which is a pro-inflammatory molecule, and is this big inflammatory response is what you see in many different allergies. They have the FC receptor, so the receptor is for the constant region of the antibody IgE, and that's where you get these responses, and they're involved in type 1 hypersensitivity
What has the FC
antibodies. The
antibodies?
IgE, the immunoglobulin E form of the bigger subclass of immunoglobulins or antibodies.
And how did they interplay with the cells? These-
Well, so if you have some sort of hypersensitivity, so let's say you are exposed to some sort of molecule, antigen, whatever, things like things like bee venom from a sting, you can get these immune responses against something that's quite mild. And what it will start causing is your mast cells will start degranulating, releasing a lot of histamine in these cases, in this hypersensitive way, and you'll get this big anaphylactic allergic reaction.
Okay.
Would you like me to talk about basophils and eosinophils?
Yes.
So these are again granulocytes. These are really for tackling your big extracellular pathogens, things like helminths or worms. Really big, kind of more associated with the gut, I would say, these really big pathogens. They're involved in your T helper 2 mediated responses. So that's where the bridge comes in with the adaptive immune response. These will be acting in the gut. They will be part of your gut-associated lymphoid tissue. But then when you get priming of your T helper 2 responses against these extracellular bacteria, pathogens, worms, helminths, or whatever, is when they will become activated really and really have a big sort of response. They are again involved in something called antibody dependent cell toxicity. Again, you can prime them with things like IgE, IgA, depends on what antibody is present, but they can start causing cell cytotoxicity and start breaking down infected cells or the pathogenic cells themselves because these big worms, helminths have quite large cells.
Priming the pathogens.
Yeah,
Okay. So now that we're on this topic, should we briefly go through the structure of the antibody?
yeah, you
Okay.
can do that. So your antibody is formed of 12 different domains. It's a protein. I think if we got this antibody up, yeah, it's got a Y shape, characteristic Y shape. Um, Sure. Yeah. So you've got your globulin domains, and you've got either a constant region or a variable region, depending on whether it can change or has different configurations. You have two heavy chains and you have two light chains, and these are held with disulfide bridges. And there's also two disulfide bridges between the two heavy chains holding them together. So the antibody will then have an antigen binding region, a Fab, F antigen, and it'll also have a constant region, FC. So this is where you get the antigen binding region bind to your pathogens. And your sentinel cells are what you really call them, your macrophages, then not really dendritic cells in these responses, but your basophils, eosinophils, mast cells will bind to this constant region, and this is how you prime with the adaptive immune system, the innate immune system, to undertake its different functions and really clear out a pathogenic infection.
Okay. And from a more chemical look, what are the variable regions or groups on the antibody?
So these are formed from recombination. You have gene segments in encoding for your T cell receptor and your B cell receptor. The T cell and B cell receptor, I'll collectively refer to them. There is variation in between them, but and they're different genes, but they both act in the same way, where it's through VDJ recombination. It's recombination of different alleles is the way that they're put together. And it's really this recombination that allows different combinations where you're able to excise with RAG1 or RAG2 recombinase enzymes to cut out different chunks, and you're able to put together different constant regions to maintain your constant region for binding to your effector cells, but also to be able to change your region, especially with further loops or CDRs on the surface, hypervariable loops, to really be able to bind to a large, diverse array of pathogens.
And now that you mentioned RAG1 and RAG2, they also play a role in T cell development. So, what are T cells? Where did they come from? Let's shift our focus to also the different types of T cells. This will be important in, are they more focused in the adaptive or the innate immune system, first of all?
All lymphoid, not natural killer cells, but your lymphoid lineage of your B cells and T cells is really part of the adaptive immune system.
Okay.
The reason I guess we'll explore it in this podcast episode is because the adaptive and innate immune system, you might think of them as two separate things, but they really act together. Where you have your initial innate responses, then you're priming with your then you prime the adaptive immune response to produce antibodies, to produce cytotoxic T lymphocytes, which will then again communicate with your innate immune cells. And what this will allow is to prime the innate immune to be able to be more efficient for things like agglutination, bringing pathogens together for phagocytosis, for example, oxidation, breaking down pathogens, which is what is really, really, what is really the effector or the mediator of breaking down your pathogens and infection. So let's look at T cells. Would you like me to how would you like me to talk about the different types of T cells, the different
Yeah, I
how they
mean,
target things, or T cell development?
I
What is the
would.
scope? Which perspective are we going to look at this in?
So you can briefly look over T cell development, but just in general, summarizing the roles of T cells and as much as is needed for us to understand the innate immune system.
Okay, so T cells are all forming from this common lymphoid progenitor. All your T cells are formed in your bone marrow, and your common lymphoid formed in the bone marrow will then travel to the thymus. This will be in very early stages of life, so in your childhood, as a baby. In your thymus, these T cells will maturate. They will start expressing the first stage is you want to be able to form your T cell receptor that is going to be complementary to multi-histocompatibility complexes. So you will have cells in your thymus with MHC1 or MHC2, and through the development of this T cell, it will either become CD8 positive or CD4 positive. So you're either your cytotoxic T cell or your helper T cell. This will come about from whether it interacts better with MHC1 or 2 because it will express both CD8 and CD4 as a naive T cell. T cells will then go through a selection process to find ones with high enough affinity to bind to an MHC complex. And this way you get rid of all the redundant ones, which would not have specificity for pathogens or your MHC restriction, because the T cell receptor binds also to the MHC. And then they will go through a process of negative selection to get rid of really high affinity binding. So if it binds with really high affinity to your MHC tissues.
What is the issue with a T cell having too low or too high affinity to certain receptors or molecules?
Too low affinity, and it will be basically useless because you want it to have affinity for your for its for its antigen on its surface. But also, if you have too high affinity, you will start binding to your own cell tissues, which is really like, looking at it from a clinical view. You have this gene called AIR, which brings in peripheral tissue. It's a transcription factor to cause presentation on your MHC of your host tissues from the periphery. Conserved cellular antigens. And if this gene is not present in a disease called APESET, then you have really big hypersensitivity autoimmune type responses and symptoms arising because you're unable to select out the T cells which can recognize your own host tissues, which is what you do not want. You want your immune system to recognize pathogens in this vast array of molecules it can bind to, but not your own host tissues, which causes autoimmunity and also hypersensitivity.
So we looked at T helper cells.
I think I mentioned the T helper 2 response with eosinophils and basophils, but there's really a whole plethora. There's many different types of T helper cells for different functions. Yeah.
We won't get into the interplay between different proteins known as interleukins, for example, in this episode. But the
Oh,
final. Do you want to?
well, I was going to mention them
Okay,
because
mention.
there is always a characteristic interleukin for response, really, for the communication to signal this type of T helper cell response. So first, you have your T helper cell priming. Now you have your dendritic cell with your antigen presentation in your lymphoid, in your lymph node. Then you will have your T cell receptor with the complement with complementary with the complementary configuration to the MHC with the peptide node. Then you have a co-stimulatory signal of B7 or CD80 on the dendritic cell and CD28 on your T cell. And what this will really do is and it, and it forces the T cell to start proliferating and differentiate. This is very important for peripheral, so you don't get excitation of T cells, and you're able to mediate only the ones with the specific antigen for it. Then you start producing things like survival factors. Interleukin 2 is the real main one,
It's an
main survival.
autocrine.
Yeah, it can act both in an autocrine way or in a paracrine way sometimes as well to meet the survival of your T cells. The T helper 1 response is really for intracellular pathogens, mainly viruses or intracellular bacteria. It's mediated primarily by interleukin 12. This will really help the survival, differentiation, and also things like recruitment of other T cells to come and be activated in the lymph node.
It can also activate natural killer cells.
Yep. And this is because this is your intracellular. This is your intracellular immune response. So you have to utilize also, there's communication here with natural killer cells to kill these redundant cells or the cells that have been infected to stop pathogen spread. It could also communicate with your cytotoxic T lymphocytes because these are also mediating the cell cytotoxicity to kill infected cells. And yeah, your interleukin 12 induces differentiation, survival, and you have your interferon gamma produced as well to activate your macrophages. Interferon gamma is the main macrophage activator. It's worth also mentioning that there's also an interferon gamma mediated positive feedback loop between macrophages and neutrophils. So you see how there's multiple different pathways and mechanisms to really amplify up the immune response for a specific type of pathogen.
What is an interferon?
An interferon is a protein and it's really important in communicating your immune response. There are interferons, there's a vast array of different, no, but interferons or interleukins.
Interferon. You mentioned interferon gamma.
Okay, so interferons are really important and are involved in your virus dependent, your viral mediated. They act to produce this antiviral state depending on what the typically they downstream, they produce downstream of your different PRRs, so your pathogen recognition receptors. So when you get a viral infection, let's say a virus is taken into an endosome, you have these receptors, your toll-like receptors most likely, and you're producing your interferons. They'll produce this antiviral state. And one of the main important things about interferons is they can recruit things like natural killer cells. To recruit an actual killer cell, that can interferon gamma is one to recruit macrophages. Then macrophages produce interferon gamma as well to stimulate natural killer cells, and it's really priming the environment to tackle a virally.
Now, a very important, we'll get into natural killer cells, but they share a very similar mechanism to cytotoxic T cells, which they release certain proteins, namely perforins and granzymes, which will act to open holes in the pores of an infected cell in order for granzymes to move in and stimulate certain molecules to perform certain functions. So specifically, a molecule called BAX will pull out BCL2, and this will stimulate cytochrome C to produce caspases that help with the apoptosis process. How do we activate these pro-apoptotic genes within the cell in when we are trying to kill the cell?
So the granzymes can have effects within mitochondria. They can have effects within the DNA. All of these are kind of starting to produce these pro-apoptotic genes. Once you get DNA damage and mitochondrial dysfunction, that's when you really start seeing this apoptotic state of the cell. With granzymes, if they go into the mitochondria and cause it to become dysfunctional, then you start leaking cytochrome C out. And it's really the cytochrome C which is embedded in the mitochondrial membrane, in a mitochondrial membrane. When you start breaking down this membrane and you start leaking cytochrome C, it's a really big signal of a damaged cell, especially if you've got that all the energy metabolism is within the mitochondria. So once you have this cytochrome C, it signals that there is damage. Your caspases become activated, namely caspase 8 or 9 with cytochrome C, especially if it's the BAX BCL2 pathway. But there's other different type of caspases that can also be activated. Range of different responses, things like the inflammasome, which again activates it's caspase 1 mediated to produce interleukin 1 beta, and all those downstream mediating effects of inflammation. But yeah, so this is that. apoptotic pathway through mitochondria, through cytochrome C, back to BCL2, having this back and forth, inducing apoptosis, inhibiting apoptosis. And this is where viruses can capitalize. You have specific viruses, let's say Epstein-Barr virus, which start producing this BCL2 homolog. So you start producing this BCL2, 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 and then it signals to not go into apoptosis when you would kind of want to. So if the cell wants to go into apoptosis, but you have these BCL2s lying around, it will signal an anti-apoptotic state, and the Epstein-Barr virus infection pathogen, the virion, will survive within the cell to replicate and then eventually cause cell death.
So there are certain mechanisms for viruses to survive. Could we mention a few?
Yeah. Well, if you'd like to tune into our other video on virology on the NeuroNXCK channel, please do so. We've done a really in-depth look at all the individual big classes of viruses and some specific examples for how they try and escape the immune system as well. But yeah, I can mention a few. There are some key ones. The main ones are things like interferon downregulation. Your interferons are trying to induce this antiviral state. Things like hepatitis C can stop the different pathways, such as your STAT2. They can inhibit these enzymes, which are part of this JAK-STAT tyrosine kinase signaling pathway that will end in transcription factors forming your interferons. Another big one is MHC downregulation. These are things by herpes viruses, cytomegalovirus, HIV. And yes, so the MHC is important for peptide loading and presenting your pathogenic material, your viral material in this case, on the cell surface, which would then kick off your cytotoxic T lymphocyte, CD8 responses. And if you can stop the presentation of damaged cells to either dendritic cells or directly to cytotoxic T lymphocytes, what you can start doing is replicating within the cell itself and kind of evading this aspect of the immune system. Now, our immune system has also found ways to come over this. If I find the right, have I included it here? No, I have not. Okay. But I hit you some of the signaling mechanisms of producing your interferon. But the way our immune system can overcome this is if viruses want to take away your MHC1 complex. The MHC1 is an inhibitory signal for natural killer cells. So if you're taking away your MHC1, you have activating signals left on the cell surface, activating your natural killer cells to start breaking down the cell. This is your missing self, which is a great way for the innate immune system to still be able to target virally infected cells and overcome this kind of viral downregulation of your MHC to evade the immune system.
Okay. So importantly, we should look at the mechanisms of natural killer cells. And first, we have to answer the question, why are natural killer cells part of the innate immune system as opposed to cytotoxic T cells?
Yeah. I don't think that, and it's not a very extensive answer, really, but it basically comes down to the fact that they're from the lymphoid lineage, but they do not present specific antigen-specific receptors. So your T cell receptor will go through all these processes to have a very specific receptor on its surface for a specific antigen, specific molecular pattern. But your natural killer cells will only have they will have receptors, but they will not be specific. So you have natural killer G2D receptor, for example. They will be able to respond to stress or damage molecules, but these are not specific to the pathogen or any kind of cellular response. It's more for a state of stress. You could have a variety of generalized signals for that.
Mm-hmm. And there are three main mechanisms as well by which natural killer cells act. You could say in a way that this means that they're not specific, they're more generalized over these three ways. So what are these three mechanisms?
Am I going to kick it back to you to tell me what they are?
Okay, so what are these three mechanisms?
So the three effective functions of natural killer cells really come from one, the perforin and granzyme release from your either missing self, if the virus is down-regulated, your MHC class 1, or your induced self, if your virus is presenting different stress signals and activating your natural killer cells, they'll start secreting granzymes perfect. Your second is what we talked about earlier with this positive feedback loop of macrophage activation. So the natural killer cell, if it's activated, will start secreting interferon gamma, activate macrophages. Macrophage recruitment will have other effective functions, but a key one is it will also secrete interferons and then start collecting in more natural killer cells. So this positive feedback loop is able to bring in more natural killer cells in this antiviral state to start attacking the cells which haven't been infected with the pathogen. Finally, you have ADCC, which is antibody-dependent cell cytotoxicity. I mentioned this briefly with your basophils, eosinophils, because they have a form of this when you have IgE from your T helper 2 responses, and you start selecting out and being able to target against really big pathogens your granulocytes. Here you have, again, you can have ADCC, where you start priming with antibodies cells that need to be damaged, need to be killed, that have been infected, and then you're able with your natural killer cell to have receptors against the constant region of these antibodies and be able to attack specifically the cells which have been infected.
By priming, you mean the antibodies binding to a certain site on the infected cell.
But the antibodies will be to a specific cellular pathogen. So if the pathogen is being presented on the cell surface with MHC1, you can start binding to that. Or if there's any danger signals present, you can also have a sort of um, adaptive immune response.
What are these MHC1 and 2 molecules?
And your MHC1, MHC2 molecules are encoded for by your HLA genes. Um, they are conserved sequences on your chromosome 6, which are able to encode for these two different molecules, MHC1, MHC2. Okay? The two different molecules come from the different responses that are being mediated through your T cells. MHC1 is presenting to T cytotoxic T lymphocytes, your CD8 receptors, and MHC2 is presenting to your lymphocytes, your T helper. So it's able to form these two different arms of your T cell responses against different types of pathogens.
Okay. So detecting the foreign antigen actually stimulates chromosome 6 to produce these C2 molecules. Yeah, okay. So is this the only, are these where they're located on chromosome 6? They prescribe both the MHC1 and MHC2?
Yeah.
Okay.
There are different genes. For example, MHC1 has HLA A, B, and C. Chromosome on chromosome 6, and then the MHC2 will be on a different locus.
Okay. Now, do we need to see the importance of dendritic cells?
Dendritic cells are important because they're what is undertaking your peptide loading. MHC are involved in presenting foreign antigen. Your MHC1 complex has a closed um, protein presenting site, and your MHC2 is open. So this brings out two different responses in antigen presentation by your dendritic cell or any other professional antigen presenting cell, which can be a macrophage or a B cell as well. And I'm just going to talk a little bit about the two different ways that a dendritic cell will start presenting MHC1 or MHC2. Because a dendritic cell has two simultaneous processes ongoing depending on what kind of pathogen it's encountering. Your MHC1 site, roughly 9 amino acids, a very short amino acid sequence because it has this receptor site. Dendritic cells are a cell, but they have very little effector function, and they react as a bridge between an adaptive immune system, going from peripheral tissues into lymph nodes and acting to present antigens to uh, T helper cells or B cells. Now, let me get back to talking to the different mechanisms for antigen presentation. Once your proteasome has broken down your pathogen, through your tap complexes, you're able to uh, move through this channel your different, the tap is a heterodimeric channel, tap 1 and tap 2. Um, you're able to move into your endoplasmic reticulum your different protein sequences. Then on your MHC1, you have calnexin, which is really a chaperone to stabilize the MHC1 complex, which hasn't been fully formed yet because you need to bind alpha 1, alpha 2, alpha 3 channel. You need to bind your beta 2 microglobulin. Okay. Then so you have this calnexin as a chaperone to stabilize your MHC1 complex. So you have alpha 1, alpha 2, alpha 3 domains on this, and you have a beta 2 microglobulin domain which you need to attach. Then once you're able to stabilize the MHC1 complex, you have a whole peptide loading complex forming around it. You have reticulin to pace it, which is a bridge path to channel in and bring in your peptides. You have ERP57 and PDI. And what these really function is to be able to bring in your peptide and load it onto your MHC. You have calreticulin, which will be able to displace your calnexin, bring in your beta 2, and then stabilize the whole structure. And then you're able to load it with ERP57 and PDI, your protein, onto your MHC. You then have ERAP to then cut off any, it is really a protease to really cut off any excess protein. So you have a very small amount of protein left within the binding site. And then yeah, you're able to form this vesicle of your MHC1s loaded with protein, and then able to present it through exocytosis to be able to fuse the membrane and express it on the surface of your dendritic cell. This is different to MHC2 in that this utilizes different vesicles. MHC2 only uses one vesicle, and once you cut up your pathogen, you have an invariant chain binding to your MHC2 binding site. This is different to MHC2 in that this utilizes different vesicular compartments. MHC2 is fully formed within the phagolysosome. In your phagosome, you're able to break down and have your different protein fragments. In your lysosome, your MHC2 complex, but it will be containing an invariant chain. This will be used to stabilize the MHC2 and stop any of these fragments just randomly binding. CLIP is an enzyme to start cutting off the invariant chain. And then you have HLA-DM, produced from this gene, which is on the HLA2 genome. Then it will remove this invariant chain and will allow peptide loading of peptide formed from the phagosome.
What is this concept of peptide loading? Where is this peptide from? What's the purpose of it?
So the peptide loading is being able to take your MHC complex and being able to put a specific peptide from the antigen of the pathogen onto it, whether it's MHC1 in your cell-mediated or MHC2 in your humoral immunity.
To attach the MHC to the pathogen.
To attach the peptide onto the MHC to be able to present it to a T cell or a B cell.
In the
And
adaptive.
in this bridge into the adaptive
Okay.
from the dendritic cell.
So, are we ready to look at macrophages?
Well, we've been ready. We've been looking at macrophages through this entire process. But if we want to elaborate a bit on macrophages, as I mentioned previously, the main the main signal for macrophages is this interferon gamma to really activate them. Macrophages come in two forms, M1 and M2, and they each have differing functions. But as a whole, macrophages are your biggest mediators of the innate system, and they have so many effective functions and signaling components that they are a critical structure, critical cell for mediating your different cellular pathways. They're involved in phagocytosis as the main one, but also they're involved in tissue repair, and they're very important for recruiting fibroblasts to start depositing new tissues through EGFR, VEGF, different epidermal growth factors, all these different mechanisms to allow them to deposit new matrix, connective tissue repair. And yeah, macrophages are really important for this communication by secreting a variety of different cytokines and signal activation of some pathways, inhibition when those are activated, like that. Fibroblasts are cell type, which is important for depositing new tissue over damage. They're important for producing connective tissue and a new extracellular matrix. You've been killing damaging cells, pathogens, whatever that's gone on through either inhibition or direct cell cytotoxicity.
What is the extracellular matrix?
It's connective tissue. As this whole meshwork of different structural proteins to hold cells together.
Okay. And what is the main process by which macrophages act?
Phagocytosis.
Process of phagocytosis.
Yeah, I think you should take this on.
Okay, phagocytosis is a form of receptor-mediated endocytosis. So an antigen will bind to a receptor on the cell surface membrane, and then a conformational change occur on certain protein subunits, which involve specific structure we look into now. Here, antigens binding to receptors trigger the extension of pseudopods, which are large projections of the surface membrane that are comprised of actin, filaments, as well as the intermediate myosin filaments. They are classed as amoeboids, and you can think of their motion as a form of pseudopodial locomotion. So, pathogens have certain conserved sequences known as PAMPs and DAMPs, with DAMPs being damage associated, which are when there's stress. PAMPs are pathogen-associated. An example of this is lipopolysaccharide on gram-negative bacteria. Now, there are certain PRRs, pathogen recognition receptors, on the macrophage surface, such as the toll-like receptor 4, which acts on LPS specifically. So this is involved in the recognition process. Now, when the antigen binds onto the cell surface, to the receptor, the recognition receptor, pattern recognition receptor, it triggers the extensions of pseudopods, which are large projections on the cell surface membrane comprised of actin filaments and other intermediate filaments, and they're classed as amoeboids. These will serve to move the particle closer to the cell surface membrane, and at this point, it will get closer and closer, and a structure of clathrin will form around these, right? It's used across all eukaryotic cells, this process. Unlike receptor-mediated endocytosis is used across all eukaryotes, unlike phagocytosis, which is specific to humans. A specific molecule known as clathrin will form around this vesicle in this kind of shape, right? This is at the point where it is fused with the membrane, this clathrin molecule will form around the vesicle to create a clathrin-coated vesicle, where this clathrin will then bud from the membrane, and it will form a clathrin-coated pit. And this coat consists of 3 heavy chains and 3 light chains, as well as an adapter protein too. Now, that's not as important. The important part is the budding. They bud to form these clathrin-coated vesicles, which then becomes an early endosome, as you can see here with the tull-like receptors. This endosome then becomes a phagosome. This lysosome will form a phagolysosome, which can then be broken down by the macrophage with acidification processes, different processes within the macrophage. And these contents can be recycled, all these things. And once we've done this, we've essentially destroyed any pathogens there. Viruses have certain mechanisms to avoid this destruction process if phagocytose into the cell. However, they also can move into the cell by other mechanisms, so they may not need to use these certain mechanisms. Okay, the important part is that the macrophages can take these, phagocytose them by this clathrin-coated process, but the membrane endosome forming a lysosome into a phagolysosome, which can then break down the contents, destroy the vesicle membrane, and destroy the contents of the cell containing the affected pathogens. This has been the second episode of the NBD, Nervous Breakdown Podcast. And make sure to check out our other channel associated with this called NeuroNXCK, which explores some of these key concepts in greater depth. The idea of this is to have a back and forth podcast covering the key aspects that most medical students and graduates, even A-level students, and even professors may be asking to look into specific processes. And we hope you have a great day.
Thank you very much.
Thank you for watching.
Please check out our other videos for a more in-depth analysis of these topics.